regions. The light chain contributes only to Fab regions. And what are the Fab? The Fab are fragments antigen binding. It determines the idiotype unique antigen binding pockets, only one antigenic specificity expressed per B cell. Now we are going to start about complement system. In complement system, the protein that are circulating in bloodstream and they can bind to pathogen, especially bacteria. These are a system of Now we are going to start about complement system. In complement system, the protein that are circulating in bloodstream and they can bind to pathogen, especially bacteria. These are a system of hepatically synthesized plasma protein that play a role in innate immunity and inflammation. They are having a various name of proteins that including that like C3, C5, C6, 3A, 3B, like it also having some numerical digits, also the combination of numerical and alphabetical digits like that. First, we are going to talk about one of the important like C3. The C3 is the most abundant complement protein and it is synthesized by liver. It's going to synthesize by liver and it's converted to C3b that is an active form. C3 inactive form that is converted into active form that is called C3b. And C3b binds to bacteria and then is called bacteria death. The all complement activation involves C3 to C3b. For the all complement activations, we have to need the process for this activation. How it is? It is like C3 is going to activate in C3b and then it will cause the death of bacteria. And they also use one component called MAC, the membrane attack complex. And guys, it's very hard for you to remember all the complement systems. Only remember this part. On the complement system is simply the conversion of C3 into C3b. And for this conversion, we are having three pathways. We are having three pathways: alternative, classical, and lectin. First, we are going to talk about alternative pathway. In alternative pathway, what is going to happen, how the activation is going to happen. So, C3 to C3b. The C3 spontaneously convert to C3b, but the C3b rapidly destroy unless stabilized by binding some components. Guys, the C3 is going to activate into C3b spontaneously, but it will be destroyed rapidly if it did not stable. Spontaneously, it converted, but it is unstable. But when it can attach or bind to some components on its surface, then it becomes stable. And how it will become stable? It can be stable by bacterial lipopolysaccharides, LPS, by fungal cell wall, and by viral envelopes. And there C3b binds amino heteroxyl group and commonly found on the surface of the pathogens. Again, I am repeating, the C3 is converting into C3b spontaneously in alternative pathway. And it is unstable. If it is unstable, it will be destroyed rapidly. But for stabilization, they have to bind with subcomponents on their surface like bacterial lipopolysaccharides, fungus silver, or viral envelope. The C3b stable can be bound by complement protein B, and complement protein D clips B bound to C3b. The forms, like what is happening, I will mark a diagram for it. It is unstable. Okay. For example, the C3b is going to bind with bacteria LPLS. It is not stable. When it will be stable, it will going to bind with some complement proteins like B and D, the protein B and D, and then it will convert it into one form that is called C3b, that's it, and C3b, capital B and D. And it is act as, it will equal to C3 convertase. C3 convertase, and this enzyme result in stable C3b can give more C3 to stable C3b. What is happening one time only? One C3b that becomes stable by joining any pathogen components, then they will be binding with the protein B and a D that will make a component C3b and Bt that will equal to C3 convertase, having the function to stabilize or convert C3 into C3b more. Like if there's a more C3 is present body, they will also going to convert in C3b by help of the C3 convertase enzyme. It is just like positive feedback. How? Like it will be activating increasing amount that will be stimulate that enzyme, and that will go and increase the amount or more. Okay. It is all about the alternative pathway. The alternative pathway is just spontaneous conversion. The alternative pathway is just spontaneous conversion. Understand? Now we are going to move to our next pathway, and next pathway is the lectin pathway. Next pathway is lectin pathway. The lectin pathway is simply the mannose binding lectin. Mannose binding lectin. The mannose is present most of our microorganisms, and that this mannose binding lectin is produced by liver, and from the liver it is going to blood and tissues. Why it is attaching a binding of mannose? The mannose is a component of many microbes. It's a bind surface with the mannose that present in many microbes, and it circulate with MAPS. Is it going to circulate with MAPS? The MAPS stand for mannose associated serine proteases. Serine proteases. And they will, what they will do, they will cleaves, convert that C3 into C2b, C4 into C4b, and then they will be joined together. The C2b and C4b is joined together and make one enzyme C2b and 4b joined together, and they will make enzyme. Already you know in the previous pathway line the C3 convertase, that is totally equal to C3 convertase. And what is going to do? They were again going to activate C3. They convert C3 into C3b. Understand? This is our second pathway. The first one, alternative pathway, in which the conveyor is going to spontaneously. The second, the lectin pathway, then it's happening by the mannose binding lectin, and in which we are cleaving C2 and C4 in 2B and 4B and making the enzyme CT convertase that doing the same step, converting C3 into C3B. Now we are going to the third pathway, and you guys know our third pathway is a classical pathway. The classical pathway is a classical pathway for the conveyor C3 to C. What is happening? They will use antibody antigen complexes. They are using antigen, antibody, antigen complexes, and they will bind with C1. They will going to bind C1, and they will cleave after binding C1. They will also cleave C2 into C2B, C4 into C4B, and the same thing will happen. The C2B and the 4B will equal to and going to activate and make like C2 activity like C3 convertase. And what is the function of C3 convertase? They will converting C3 into C3B. Understand? Again, there are three pathways for the activation of C3. First is alternative, second is lectin, and third is classical. In alternative, they are going spontaneously. In lectin, there is a mannose binding lectin, and in classical, they are using antibody antigen complexes for the activation of C3 into C3B. And what are important enzyme? C3 convertase. That is understand for you guys. Hope so. And we are talking about C1. The C1 is a large complex. We talk about the C1. The C1 is a large complex. And we'll draw its structure. It having the structure of C1Q. Okay. Having the structure of C1Q, C1R, C1S, and having one C1 inhibitor. Is a structure. Normally it is inactive due to C1 inhibitor attached with it. But it must bind to two FC portion close together for the activation. What we're going to happen, for example, if there's know about the IgM, it having 5 sites for binding with the antibodies. Like it's 1, 2, 3, 4, and 5. Structured like that. Understand? And you guys also know there is a lower part, the FC part. The FC region belongs to complement binding. Complement binding. So if the two FC bindings are close to each other, okay, the C1 is going to bind with them and they are too close to each other, then it will be going to activate. And by that, the C1 inhibitor will fall off and it will activate. How? Because the inhibitors are removed and fall off, and then the C1R and C1S become more active or active, and they will also create same thing, C3 convertase. And what is happening for them? In which pathway it is happening? It is happening in classical pathway. Classical pathway for the activation of complement system, in which we are using antibody antigen complexes. Understand? Well, we were talking about the activation by spontaneously they will attach and staple in alternative pathway, but how our own body cells, how the host cells are protected from the C3B. It is protected by one factor and its name as factor H. What is going to happen in the factor H? The factor H is a plasma glycoprotein also synthesized by the liver, and it blocks the alternative pathway, on host cells. What is happening in alternative pathway? The C3 is converting the C3b spontaneously, and it is unstable. To getting stable itself, it needs some components. I'm right? It needs some components like amino or hydroxyl group that also present in host cell, but host cell protect it by some factor, that is factor H. And what is it doing? It accelerates the decay of C3 convertase. Factor H is going to accelerate decay, decrease the amount of C3 convertase, that a more common component for the conversion of C3 into C3b. And secondly, it cleaves and inactivates C3b itself. Like that. No entrance for the host. Used by the cancer cells, exactly haven't no, like for example, if I am a doctor, I am using a medicine for some component, I also know how to use and how it will be outcome for when I have to use to protect myself. And now this thing, the factor H mechanism is also going to copied by our enemies. Who are our enemies? Like cancer cells and some bacteria. They know the secret of factor H, and these also taking and adopting the same mechanism to protect themselves from C3b. You guys